So today we are going to discuss carbohydrate digestion. Now listen, before we proceed with carbohydrate digestion, you should understand that what is the most unique thing about carbohydrate digestion. Can anyone say this? Unique thing about carbohydrate digestion is carbohydrate? Okay, no. Listen, the unique thing about carbohydrate digestion is this: you need to understand that it is one of the type of digestion in which the digestive enzyme is not at all cleaved. That means the active enzyme is secreted in its original form. Okay. Now listen. When we talk about carbohydrate digestion, we will be talking about actually monosaccharide, okay, oligosaccharides, and polysaccharide. Okay, monosaccharide, oligosacchariide and polysaccharide. Now listen, before we start this, we need to understand that what do you mean by monosaccharide, oligosaccharide and polysaccharide. Before we continue this discussion, we should understand that what is monosaccharide, oligosaccharide and polysaccharide. Now listen, we've already discussed in great details these things when we were discussing carbohydrate biochemistry and glycolysis Krebs cycle. Now listen, so what is monosaccharide? Monosaccharide means single glucose or single molecule, right? It's a single molecule thing. Now listen, when we talk about oligosaccharide, what do you mean by oligosaccharide? Oligosaccharide means two or more, right? Now listen, for digestive purposes we will know a different definition. Oligosaccharide means short polymers. Okay, polysaccharide means long polymers. You know polymers? No. Okay, so short polymers or long polymers? Polymers are what? Polymers are actually made up of repetitive units. Polymers are actually made up of repetitive units. Now here the repetitive unit is in most of the cases glucose. In most of the cases this repetitive unit is actually glucose. Okay, now we will understand step by step. Okay, but first you should understand a very basic thing. What is our aim actually? What is our aim? Now listen, our aim is to convert polysaccharide and oligosaccharide. Our aim is to convert polysaccharide as well as oligosaccharide into monosaccharide. Okay, our aim is to convert oligosaccharide as well as polysaccharide into monosaccharide. Clear? Okay, now listen with all your attention. You should understand a very basic concept, that why we are talking about that body will have to convert polysaccharide as well as oligosaccharide into monosaccharide. This is because of the fact that body only absorbs monosaccharides, not polysaccharide or oligosaccharide. Clear? Is it okay? Okay, now listen. There are basically three monosaccharides we will talk about today. Three monosaccharides whose digestive process we will talk about. Okay, that is glucose, the central of them all. Next is fructose. Third is galactose. Okay, first is glucose, second is fructose, third is galactose. Clear? Now listen, whenever we talk about carbohydrate, okay, you should always understand, in most of the case carbohydrate comes from plant sources, not animal sources. In most of the cases there are some animal sources like glycogen, okay, but in most of the cases we take carbohydrate in the form of starch, okay, which is a plant. What is the exact definition of starch? What is the exact definition of glycogen? It's called animal starch, okay, but what is the exact definition of glycogen? Glycogen is a highly branched and storage form of glucose in our body. That means whenever you take suppose 100 glucose, 80% of the glucose will be converted into glycogen. That means body will store this glycogen or this particular glucose in the form of glycogen so that it can use it in near future. Okay. Now in plants, plant also have this strategy, you know, to store carbohydrate or to store glucose, and they produce, instead of glycogen, starch. Okay. Now listen. So our main purpose is to know how these two get digested and are converted to this monosaccharide. Okay. Now listen. We will concentrate mainly on two things, that is today we will know that what are the ingested main ingested carbohydrates which we take as food. Number one is starch. Okay. Number two is sucrose. Sucrose is also known as table sugar. Okay. Number three is lactose. When we will talking about digestion of carbohydrate, actually we are talking about the digestion of these three: starch, sucrose and lactose. Clear? Okay. Now listen. In the second column, we will talk about digested products. Okay. That means in which form, these are converted into which form? Listen. Starch, sucrose and lactose, they are all converted into three basic things. Number one, glucose. Number two, fructose. Number three, galactose. Okay. So our body actually extracts glucose from starch. Our body extracts glucose as well as fructose from sucrose. Our body extracts galactose from lactose. Okay, because lactose is actually a dimer of what? A dimer of disaccharide. It's a dimer of what? Glucose and galactose. Clear? Okay. Now listen. There are many biochemical views about this. Now listen with all your attention. First of all, we should understand that how, body, take care of these things. Listen. Starch is the main, starch is the main form of glucose in our body. Okay. And listen with all your attention. Starch is the main form of glucose and is the longest form of glucose. Now listen. When we are talking about digestion of carbohydrate, you should always understand that when we are talking about digestion of carbohydrate, we are actually talking in general about starch digestion. Why? Because in books it is said that carbohydrate digestion starts in the mouth, right? Clear? Now you should understand very clearly that in the mouth actually starch digestion starts. Sucrose and lactose digestion doesn't occur much in your mouth. Clear? Because salivary gland do not secrete sucrase. Some evidences are there, but they are not actually good evidences for us to understand that it is secreted by your mouth salivary glands. Okay, clear? Now we will understand today that how starch is digested in our body, how sucrose is digested in our body, how lactose is digested in our body, and in the next lecture we will study that how the digested products are actually absorbed into the cell, okay, of GI tract. Now listen with all your attention. So first step of carbohydrate digestion, listen. In many books and in many lectures you will find out that some people are saying that carbohydrate digestion when salivary amylase comes out. Listen, there are two ways you can approach carbohydrate digestion. Number one, by sight, that means you can study like carbohydrate digestion in mouth, carbohydrate digestion in intestine, that is small intestine specifically, or you can study by using the famous technique of biochemistry, that is individual studying of each and every enzyme. But listen, which one is easy? We will obviously get into the details of biochemical structure, extracrystallographic structure of salivary amylase as well as salivary amylase or tyalin and pancreatic amylase. Don't worry, we'll get into the details, don't worry. But we must understand, we should approach with respect to sites, that is carbohydrate digestion in mouth, then in the small intestine, then in other places. Okay, so we will at first talk about carbohydrate digestion in mouth. Okay, now listen. In mouth, carbohydrate digestion occurs by the help of by the help of salivary alpha amylase. Salivary alpha amylase. Okay, now listen, this is the this is salivary amylase. Okay, this is the writing. Okay, now listen, now you should understand the basic thing, that why salivary amylase is so much active and so much important. Now listen, you should understand that salivary amylase, which is also known by the name of tyalin, tyalin is actually common name for salivary amylase. Now listen, this is suppose chromosome 1. Okay, this is suppose your chromosome number 1. Okay, now listen, in chromosome number 1, in the short arm of chromosome number 1, okay, in the short arm of chromosome number 1, there are two genes for amylases, AMY1 and AMY2. Okay, AMY1 and AMY2. Okay, now listen, AMY1 give rise to tyalin or salivary alpha amylase, and AMY2 give rise to pancreatic amylase. Now listen, first I will discuss the structure of salivary amylase, then I will actually put some extra information with respect to pancreatic amylase. That's why you will know that salivary amylase only, remember this for your entire life, salivary amylase only acts on boiled starch. Okay, that means rice or something like that which is being boiled. Now listen, if you take raw grain or raw rices, suppose, it will be taken up by pancreatic amylase, that is acts on both boiled as well as unboiled starch. Okay, that's the difference. Now listen, now, so you now know the difference between pancreatic amylase and salivary alpha amylase or tyalin. Now we should draw the peptide of protein of salivary alpha amylase, and then we will understand the difference between pancreatic amylase and salivary amylase. Now listen, suppose this is the protein which is transcribed and translated from AMY gene. Okay. Now listen with all your attention. This protein has three main regions. Okay. The first region is called A region. A region. And the second region is called B region, and the third region is called C region. Now listen, this A region, to this A region actually this A region this A region is associated with C terminal of ptyalin, and this C region is actually associated with N terminal. Every protein has an N terminal and C terminal. Okay. Now listen with all your attention. You should always understand this. Now listen. This is the main catalytic region. Okay. The C is the main catalytic region where bonds are broken, and now listen, what are the uses of these two regions? No need to understand that, no need to memorize this, but just for the information sake you should remember this. That is, A region stands for activation site. That means whenever this particular portion is cut off, then this whole protein will be native. That means no work will be there. That means A region is the activation site. That means if you activate this site, then the whole protein gets activated. Now, how can we activate this region? By introducing chloride. Chloride activates A region, which is frequently present in our food. Okay. Now, the B region contains calcium as cofactor. The B region contains calcium as cofactor. What is the importance of B region? B region helps in configuration of this protein. That means correct configuration of salivary amylase is actually the function of this B region, and this C is the catalytic domain. Catalytic domain. Okay. So this is the structure of salivary alpha amylase. Okay. This is the structure of salivary alpha amylase. Okay. The structure of salivary alpha amylase. Now listen. You should always understand that I have written here that salivary amylase acts only on boiled starch and pancreatic amylase acts on boiled as well as unboiled starch. Now, why this difference? Why this difference? Why raw starch cannot be handled by pancreatic amylase? Sorry, salivary amylase. The main important thing is this: you need to understand that how many domains are there in salivary amylase? There are actually 3 domains, right? A, B and C. Now listen, in pancreatic amylase there are actually 5 domains. These extra 2 domains are actually associated with this special task of handling unboiled starch. Okay, these extra 2 domains are actually dedicated to handling unboiled starch. Okay, clear? Okay, so by far we have discussed the chromosomal, this is chromosome 1, okay, this is chromosome 1, that is chromosomal, presence as well as the structure of salivary amylase. Now we will talk about how this salivary amylase acts on food like starch. Now listen with all your attention. Before we start this, we should also understand that how our body senses any chemical activity in our mouth and triggers the action of salivary amylase or triggers the salivary gland to release what? Salivary alpha amylase. So now listen. Suppose this person is actually have taken a food bolus. Okay, now listen with all your attention. Now listen, suppose this person has taken a food bolus, okay? Now, when you are chewing, you know, suppose this is your salivary gland, sublingual salivary gland, okay? Now listen with all your attention. Suppose this is your salivary gland, okay? Let me draw it here. Now listen. Suppose this is your salivary gland, you know, salivary glands are acinar glands. That means they have acini, bind sac-like structures which are, you know, cellular, which have cellular lining. Okay, clear? Okay. So these are your acini. Now listen. Okay. So this is one of the, you know, magnified version of this salivary gland. Okay, this is one of the magnified version of this salivary gland. Clear? Now listen. Now whenever I am talking about salivary gland secretion, okay, or salivary secretion, you should always understand few basic things. Suppose your dad or mom is scolding you, or your girlfriend or boyfriend is scolding you, suppose. Have you noticed this one thing, that you know, the water content of your mouth decreases suddenly and you feel like drinking some water, but probably you have drank water just few minutes back. Why this happens? This is because of the fact that whenever you are getting a scolding from anyone who is close to your heart or anything else, okay, this is because whenever you are getting a scolding, your sympathetic system gets activated. Okay, gets activated. Now listen, always remember this: sympathetic starts with S and it is associated with stress. That means whenever someone is scolding, do you think it's a very comfortable situation for you? No, it's obviously it's a stressful situation. That's why sympathetic system will fire. Sympathetic system will fire. And you know what will happen? You know what will happen? When they will fire, they will release acetylcholine here. Okay, sorry. Adrenaline. Sorry, adrenaline. No. Why I was telling sorry? You know why? Because in sympathetic system, there are two neurons. Okay, one will, you know, come out from central nervous system, which always secretes acetylcholine. Any neuron coming out of central nervous system secretes acetylcholine. And number 2 point is this: you know, to understand that sympathetic system, in the second neuron of sympathetic system, suppose this is the first neuron. First neuron of sympathetic system always releases acetylcholine. The second neuron always releases adrenaline. But you should always understand you should always know the exception. The exception is sweat gland. In case of sweat gland, both the preganglionic as well as postganglionic neurons secrete acetylcholine, though they are sympathetic. Okay, they secrete acetylcholine. That's a difference. Okay, now listen. So this will. So what will happen? This neuron which is coming out. So this is what? This is actually the postganglionic neuron. This is actually what? Postganglionic neuron. Okay, now listen. Actually the postganglionic neuron now activates or fires, and as adrenaline is released. Now listen. These cells, now listen, there are two types of cells present in salivary gland. These cells which surround the acini are called acinar cells. Acinar cells. And these cells which, you know, surround the tubes, okay, these are called tubular cells. or this is also known by the name of duct. That's why they are also known as ductal cells. Now listen. Postganglionic neuron of sympathetic nervous system innervates acinar cells. Okay, they innervate acinar cell. Now I will be drawing a big, enlarged version of acinar cell. Suppose this is an enlarged version of acinar cell. Now listen with all your attention possible. Okay, now they will secrete adrenaline, right? And adrenaline is what? Adrenaline will bind with what? Adrenaline will bind with beta receptor. Okay, and they will lead to the high concentration of cyclic AMP. Okay, GS signaling. That's it. G protein coupled receptor signaling, GS. Okay, and this cyclic AMP will lead to the activation of protein kinase A, and protein kinase A will inhibit all the proteins which will help in the secretion of anything or saliva. Okay, now listen. What happens? Now listen with all your attention. The problem is this, to understand that whenever you are in stress, sympathetic nervous system fires and postganglionic neuron, secretes adrenaline, and adrenaline is associated with beta receptor. Adrenaline binds with beta receptor present in the acinar cells, and that lead to increasing cyclic AMP concentration. As they lead to the increased cyclic AMP concentration, therefore what happens is actually, so now listen. Suppose you are in a happy-go-lucky mode. That means what? You are relaxing. Okay. When you are relaxing, okay, parasympathetic activity goes up in your body. Now listen. Whenever you are relaxing, parasympathetic activity goes up in your body. Okay, what happens? In parasympathetic activity, the preganglionic as well as the postganglionic neurons both are cholinergic. That means release acetylcholine. Okay, both are cholinergic. Now listen, when we are talking about parasympathetic system, you should always understand that suppose this is the postganglionic neuron. Okay, suppose this is the postganglionic neuron. Okay, now here acetylcholine is released. Now this is muscarinic receptor. Muscarinic receptor. Now listen, muscarinic receptor is actually coupled to GQ protein. Okay, GQ. Muscarinic receptor is actually coupled to GQ protein, and GQ protein leads to what? Activation of phospholipase C beta, and then IP3 and DAG, right? Now IP3 leads to the increased calcium level in your cell, and that leads whenever increased calcium level occurs in your cell, then remember this, in most of the cases some secretion will be there from the cell, either neurotransmitter release or secretory vesicles will be released. Now these secretory vesicles contain salivary enzymes, and these salivary enzymes or salivary alpha amylase is released from this. Okay, now listen. Now point you need to understand is this, that salivary secretion increases when parasympathetic activity is there. Salivary secretion decreases when sympathetic activity is there. Okay, now listen. Okay, so that's it. Now we have already understand that how salivary enzymes are secreted. Okay, now we'll talk about what happens when these salivary enzymes are secreted. Now listen with all your attention. I should say another thing, that salivary alpha amylase is one of those enzymes which are secreted in the active form. unlike protein digestive enzymes which should be cleaved, that means propepsinogen, chymotrypsinogen, trypsinogen. But salivary alpha amylase is an enzyme, and even pancreatic amylase is an enzyme which is secreted in their active form. Okay, clear? Now you should always understand that what is the basic difference between salivary alpha amylase and pancreatic alpha amylase. Now listen, there is a basic difference that both acts on boiled starch, but salivary alpha amylase never acts on unboiled starch. But pancreatic amylase is far more better because it acts on both boiled as well as unboiled starch. And there is another difference between the difference between salivary alpha amylase and pancreatic alpha amylase. You know what is that? The basic difference is this: salivary alpha amylase works in a neutral or slightly acidic pH. But listen with all your attention. But pancreatic amylase acts on alkaline pH. That's a very basic difference. Salivary amylase acts on what? Salivary amylase acts on slightly acidic or neutral pH, that is 6.7 pH. pH 6.7 is the ideal for salivary amylase. But pancreatic alpha amylase acts on alkaline pH. Okay? Now listen. Suppose now we will study the effect of salivary alpha amylase on starch. So this is the best part. Why? Because whenever we are talking about carbohydrate digestion, you should always remember in most of the cases this actually predominates. Okay. Okay, so we were discussing effect of salivary amylase on starch. Now listen, before we discuss, we have already discussed the starch structure of starch, but you should understand that it's very high time for us to understand again the structure of starch. Now listen, starch is made up of two units, right? That is amylose and amylopectin. Amylose and amylopectin. Now listen, what is amylose? Amylose is a straight chain structure. Straight chain structure. And amylopectin is a combination. Now listen with all your attention. I have already discussed this. Combination of straight chain as well as branched chain structure. Okay, clear? So this is the basic difference. Now listen, so starch is actually made up of two things, amylose and amylopectin. Okay, now we will talk about how this amylose and amylopectin differ. Now listen, if I say you know already, that's why I am saying you directly, that amylose contain alpha 1, 4 linkages, and if I say amylopectin contains both alpha 1, 4 as well as alpha 1, 6, you know already, but we will have to discuss the structure of starch. Why? Because salivary alpha amylase have 3 basic limitations. That's why it cannot properly digest starch. Yes, salivary amylase as well as, now listen with all your attention, salivary amylase as well as pancreatic amylase cannot convert starch into glucose. They can never. Why? Listen, both have serious problems with respect to starch structure. Listen, Suppose this is a glucose molecule, okay? Suppose this is a glucose molecule, okay? This is carbon number 1, this is 2, 3, 4, 5 and 6, okay? Now listen. Suppose this is another glucose molecule. Okay, this is carbon number 1, 2, 3, 4, 5, 6. Okay, now listen. Suppose here is OH, here is OH. Now listen. Suppose there is a glycosidic bond formation here, and this will come out as this. Now listen. In this glycosidic bond, this is a glycosidic bond, right? This is a glycosidic bond. This is a glycosidic bond. Now listen. In this glycosidic bond, this is the first glucose and this is the second glucose. Okay. Now listen. In this glycosidic bond, you should always understand that the first carbon of the first glucose and the fourth carbon of the second glucose is actually taking part, right? That's why it is called, this glycosidic bond is called 1,4 glycosidic bond, okay? Now, we have another term that is alpha. Why it is called alpha? Because both these, listen with all your attention, both these glucose are alpha isomers, sorry, alpha anomers. Both these glucose are actually alpha anomers. Okay, that's why they are called, that's why we put an alpha mark here. Okay, clear? Clear? Now listen with all your attention possible. That is, we put an alpha mark because both these glucose are alpha anomers. We put 1,4 here because the first carbon of the first glucose and the fourth carbon of the second glucose is actually taking part in the formation of this glycosidic bond. Okay, now listen. Now I will be drawing the structure of, a basic structure of starch, and in this right corner I will tell those three important points which you should remember when you are talking about salivary amylase. Now listen, give me a minute so that I can draw that. This is alpha 1,4, right? This is alpha 1,4. This is alpha 1,4. Okay, this is alpha 1,4, right? Okay, now listen, this is alpha again 1,4. Now listen, this is alpha 1,6, first carbon and the sixth carbon of the second glucose. Okay, and again we continue our journey. Alpha 1,4, alpha 1,4. Okay, now listen. So this portion can be considered as amylose portion. that is contains only alpha 1 comma 4, but this portion may be considered as alpha 1 comma may be considered as amylopectin. Okay, clear? Okay, now listen. Amylopectin contains both alpha 1 comma 6 as well as alpha 1 comma 4, right? Now listen with all your attention. This is the alpha 1 comma 6, this is alpha 1 comma 4. Okay, this is alpha 1 comma 4, alpha 1 comma 4, alpha 1 comma 4. Okay, now you know all these things. Now listen with all your attention possible. Listen. So you have seen the structure of, a bit small structure of, glucose, starch. Now listen, in the amylopectin structure, amylopectin is branched structure. Amylopectin is branched. Okay, but amylose is a straight chain structure. You can now see. Now listen. Why amylose is a straight chain structure? Because it contains only 1 comma 4, right? Contains only 1 comma 4, alpha 1 comma 4 linkages, right? Okay. Oh, there is a very big difference. He was saying that. So what is the difference in starch and cellulose? Both are plant derived, right? So what is the difference? Now listen. Starch contains alpha 1 comma 4 and alpha 1 comma 6, right? Cellulose contains beta 1 comma 4 linkages. That's a very basic difference. Okay, they contain the beta anomer of glucose. Okay, that's why it cannot be digested. Listen with all your. That's why we human beings cannot digest cellulose. Why? Because we don't have any salivary beta amylase or pancreatic beta amylase. Clear? That's why cellulose acts as a roughage. Okay, cellulose, lignin, pectins, these all act as a roughage. Okay, now listen. Now, in this structure, we will actually talk about the action of salivary amylase, but there are three basic things you need to understand. There are three basic important things you should understand when you are dealing with salivary alpha amylase. Three things are: number one, salivary alpha amylase never acts on terminal alpha 1,4 linkage. That means whenever, suppose, never acts on terminal alpha 1,4 linkage, never acts on terminal alpha 1,4 linkage. That means salivary amylase will never act here. It's a very much vulnerable position, but it will never act here. It will never attack this position. Why? Because it is terminal. Okay. Number two, you should understand, salivary alpha amylases never acts on alpha 1,6 linkage. That means salivary alpha amylase will never attack here. Clear? They will never attack here. And the third and the most important thing, salivary alpha amylases never acts on linkage just before and after alpha 1,6. Now listen, this point you should understand in detail. Now listen, this is alpha 1,6 linkage, right? This one is alpha 1,6 linkage. Now this is the linkage of alpha 1,4. Okay, but this alpha 1,4 is just before the alpha 1,6 linkage, and this also, this alpha 1,4 linkage is also what? After alpha 1,6 linkage. So you should always remember, in these positions, listen with all your attention. In these positions, no action of salivary amylase will be detected. In these positions, no action of salivary amylase will be detected. Clear? Okay. Okay. Now Now we will try to understand, suppose this is another, I am giving it for my own benefit, okay. Suppose this is another, alpha 1, 4 linkage, and this is another alpha 1, 4 linkage. Okay. Now listen. Now point to understand that, listen. So this is actually this one, okay. Now listen with all your attention. So these, terminal alpha 1, 4 linkages, suppose we take this thing in a test tube and we put salivary alpha amylase. Now listen. Now these terminal positions will be okay. Now I am starting from this, part. That means we will see from this part, we will start from this part, that what are the products of salivary amylase digestion. Now listen. So this is the terminal position. Salivary amylase will not work here. That means it will go to the second one. That is this. This is the vulnerable position. Here it will cut. That's why the first product of salivary amylase we are getting is maltose. Okay. Clear? Now listen. There was another chance, that salivary amylase before cutting this area will act here. If it acts here, it is called, now listen, if it cuts here, this is called maltotriose. Okay. So two products we are getting directly, that is maltose and maltotriose. Okay. Starch digestion product. Now listen. Okay. Now you need to understand, suppose this is, the position, and this is the position where salivary amylase can work. So here you will get a zigzag, thing, that is this one. You will get a zigzag thing like this, right? Right? Okay. So this structure, this whole structure is actually not touched by, salivary alpha amylase. So as well as pancreatic amylase. Now listen, this structure is called alpha limit dextrin. Okay, this is alpha limit dextrin. So what are the digestive products of starch? Okay, that is alpha limit dextrin, maltotriose and maltose, and maybe if you are fortunate enough, you will get one or two among glucose residue like this. Okay, now listen. You should understand now that you are already seeing these things, that suppose this is maltose. Maltose has this alpha 1, 4 linkages, and maltotriose has two alpha 1, 4 linkages, suppose. Okay. Now listen with all your attention. You should understand now that you can very well understand that this is a what? This is an oligosaccharide or short polymer. Okay. This is an oligosaccharide, right? Now listen. So salivary amylase as well as pancreatic amylase actually converts polysaccharide or long polymers into oligosaccharide or short polymers. But what I have said in the first session is this: the main important point about carbohydrate digestion is this: we will have to convert long polymers or polysaccharides as well as short polymers or oligosaccharides into monosaccharides. Do you think these are monosaccharides? No, they are oligosaccharides or disaccharides. Alpha limit dextrin is a oligosaccharide. Again, maltose is a oligosaccharide. Now listen. So we have actually converted a polysaccharide into oligosaccharide. Now our next step is to convert oligosaccharide into monosaccharide. Okay. Now we'll convert that now. Listen. This conversion occurs in the tip of jejunum. Okay. That is conversion of oligosaccharide into monosaccharide. This conversion occurs in the tip of jejunal cells. Okay, suppose this is a jejunal cell. Okay. Suppose this is your duodenum, then it is your jejunum, suppose. Now listen with all your attention. This is jejunum and this is suppose jejunal cell. Now listen, I am drawing a bigger version of jejunal cell here. Okay, so this is a jejunal cell. Now listen with all your attention. You should understand, in the jejunal brush border there are enzymes fitted to the lipid membrane of cell membrane, okay, lipid bilayer. Okay, now listen, there are three main enzymes associated with carbohydrate digestion in jejunum. These three main enzymes are number one, lactase, number two, sucrase, okay, number three, number two, sucrase isomaltase, third one is maltase. Okay, so these are the three enzymes. Now listen, we'll fit here, okay, suppose this is the now listen, suppose this is called lactase, okay. Now listen, lactose is actually milk sugar. That means when you are taking in milk, then lactose goes into your body, and what happens? Listen, in lactose there are two units, one is glucose and another is galactose. Now listen, this lactase is an enzyme which actually is present in the luminal brush border of jejunal cell, and it is present as an integral membrane protein of these cells. Okay, now listen, that means they are attached to the cell membrane. Now listen, when lactose reaches jejunum, this enzyme acts on them and convert them into glucose and galactose. Okay, clear? Now we'll talk about the second enzyme, that is sucrase isomaltase. Why these two names are there? Now listen, these two names are there because the structure of this enzyme looks like this. Now listen, this is the sucrase part, this is the isomaltase part. Okay, now listen with all your attention. This enzyme is actually two enzyme or double enzyme. That means they are having an isomaltase part, they are having an sucrose part, and they are forming a dimer also. Okay, and they are attached to the luminal brush border epithelium. Okay, now listen, when sucrose comes, it is converted by this domain into glucose and fructose. Okay, glucose and fructose. Now listen with all your attention possible. Isomaltase is actually the domain or the enzyme which converts alpha limit dextrin. I have already discussed the structure of alpha limit dextrin, which converts alpha limit dextrin into glucose residues. Clear? Okay. Another thing we need to add here is what? Maltase. What is the function of maltase? You already know, right? Now listen, this is the structure of maltase. Okay, now listen, this maltase again is an integral protein, converts maltose or malt sugar into 2 glucose units. Okay, so this is how your body, converts every possible carbohydrate oligosaccharide into monosaccharide. Okay, so there are basically 3 enzymes which are present in the jejunal cell apical surface, and they are present as an integral membrane protein of the jejunal cell surface, jejunal cell membrane. Okay, clear? Okay, let's take a break for some time, then we will talk about the absorption of these monosaccharide units into the cell. Okay, thank you. So now we will be discussing digestion of carbohydrates in small intestine. Now listen, whenever we are talking about small intestine, it is also called pancreatic digestion. Okay, it is also known by the name of pancreatic digestion because pancreas plays a major role in this type of digestion. Now listen, now so in stomach what we have read is actually, listen, in stomach what we have read is actually here in the mouth salivary alpha amylase converts starch into alpha limit dextrin, maltose, maltotriose, etc. Now listen, this whole component, that means maltose, maltotriose plus alpha limit dextrin together with salivary amylase now enter stomach. Now listen with all your attention. In stomach the pH is really low, right? And at low pH salivary amylase stops working. Clear? That means in stomach there is no digestion of carbohydrate. Now listen, when low pH means what? Low pH means high concentration of H plus. Now listen, when this H plus will reach small intestine, when this high H plus concentration or these H pluses will reach small intestine, what will happen, you need to understand is that suppose this is your intestinal cell and this cell have a mechanism to sense H plus concentration. Okay, and this H plus when enters this cell, okay, these cells will actively secrete secretin. Okay, now listen, now one thing you need to understand again is this, that is when food and other particles reaches the small intestine, then when food and other particle reaches the small intestine, then you need to understand is this, then vagus nerve also gets activated. Okay, vagus nerve also gets activated. What is vagus nerve? Vagus nerve is nothing but a parasympathetic nervous system part. Okay, now when vagus nerve gets activated, okay, now what will happen is this, you need to understand now. Listen, suppose here there is pancreas. I am just drawing pancreatic duct, one pancreatic assignee, and this is your pancreas. Okay, suppose for the time being just understand this funny thing as your pancreas. Okay, clear? Suppose this is your pancreatic duct, sorry, this is your pancreatic assignee, this is your pancreatic duct, and this is your pancreas. Now listen, vagus nerve now activates pancreatic acinar cell. Now listen, vagus nerve activate pancreatic acinar cell. Now listen, when pancreatic acinar cells are activated, suppose this is a pancreatic acinar cell and vagus nerve activate this, vagus nerve will act is a what? Vagus nerve is actually a parasympathetic extension. That means it will secrete acetylcholine, and this acetylcholine is bound to what? Muscarinic receptor. Okay, and this leads to high concentration of IP3 and DAG. Okay, that means phospholipase C is activated and IP3 and DAG is produced, and due to IP3 calcium will be released and some vesicles will be released due to the release of calcium, and this vesicle contains the enzymes. This vesicle contains the enzymes for digestion. That means chymotrypsin, trypsin, chymotrypsinogen, trypsinogen, and all other enzymes like pancreatic amylase as well. Now listen, suppose this is your pancreatic acinia and this is your pancreatic ductal cell. What is the difference? There is a huge difference between pancreatic ductal cells and what is the important anatomical position here? Whenever you are given a slide where you will have to select that whether you are seeing the sample as or the slide as pancreas or salivary gland, you should understand that pancreatic ductal cells approach the acini and they enter the acini. But salivary ductal cells will never approach or will never encroach the salivary acinar cell. That means salivary ductal cells will never encroach or enter what? The salivary acini. But pancreatic acini is actually invaded by ductal cells. Okay, that's a very basic difference. Okay, these ducts enter, pancreatic ducts enter the what? Pancreatic acini. Now listen, what is the basic difference in ductal cells and salivary pancreatic acinar cell? Pancreatic acinar cells secrete the enzymes. They secrete the enzymes in active or in inactive form. But pancreatic acinar cells secrete HCO3 minus. Why? Because these cells contain receptor for secretin. These cells contain receptor for secretin, and secretin activates a protein which actually throws away HCO3 minus and takes in chloride. Okay, which actually throws away HCO3 minus and takes in chloride, and this HCO3 minus actually when comes to small intestine neutralizes H plus coming from stomach, and therefore when excess amount of HCO3 minus is released, then you should understand that when excess amount of HCO3 is released, then you should understand then and there that neutralization of H plus has occurred. Now excess amount of HCO3 means the whole medium is now basic, and in the basic medium pancreatic amylase can work. Now listen, you people have a question, that is what he was actually asking is HCO3 comes from an acid, right? HCO3 comes from, suppose, H2CO3, carbonic acid. Then why after breaking down HCO3 becomes a base in our body? It's a base. Why? Because HCO3 is a weak acid. HCO3 is a weak acid. Okay, and the concept of conjugate acid base pairing, conjugate acid base, what is that? Listen, it says that whenever a weak acid will liberate its H plus, the all part which is remaining after the liberation of H plus will be the opposite of this acid. That means weak acid, when it is giving out H plus, have a strong base. A strong base. Why HCO3 is a strong base? Because of resonance. Okay, as it is a strong base, therefore liberation of HCO3 in the pancreatic, liberation of HCO3 by the pancreatic ductal cells is an important step for the action of pancreatic enzymes to work. Okay, because pancreatic enzyme cannot work in acidic pH. Okay, Clear. Now we should understand one thing, that what is the basic difference between pancreatic amylase action and salivary amylase action. Listen, we have already studied in great detail the salivary amylase action, right? Salivary amylase performance. Now listen, there is no difference in the performance of salivary as well as pancreatic amylase. The only difference is this: it works in alkaline pH, it works in slightly acidic pH or neutral pH. The second difference is this: it acts on boiled starch only, but pancreatic amylase acts on boiled as well as unboiled starch. Why we have already discussed, because of two extra domains. Okay, that's it. Pancreatic amylase, starch when acted upon by pancreatic amylase will also give alpha limit dextrin, maltose, maltotriose. That's it. There is no difference. Okay, clear. Before taking the break, we have discussed in great details the carbohydrate digestion. Okay, now we'll talk about, just for few minutes, carbohydrate absorption. Listen, carbohydrate absorption actually starts in the jejunum itself. Okay. Now listen, suppose this was your jejunal cell. Okay. Now listen, a jejunal cell has three proteins: the lactase, okay, sucrase isomaltase, right, which is a dimer, right, sucrase isomaltase and maltase, which is also known by the name of glucoamylase. Okay, glucoamylase. Okay, now listen, upon reacting with these three, alpha limit dextrin was converted into glucose, lactose was converted into glucose and galactose, and maltose plus maltotriose was converted into glucose, and sucrose was converted into glucose plus fructose. Right, now listen, so which is the most abundant monosaccharide which we are going to learn first with respect to carbohydrate absorption is glucose. Why? Because you can see in most of the cases glucose is the product produced. Now listen, for the absorption of glucose you need to understand that two mechanisms or two steps are involved. Okay, now listen, there is a protein here, okay, in the cell surface membrane, okay, cell membrane, there is a protein by the name of SGLT, or sodium dependent glucose transport. Okay, SGLT. To be very specific, this is SGLT1. Okay, now listen, now you need to understand one basic thing, that SGLT1 or sodium dependent glucose transporter number 1 will actually help in the absorption of glucose, but to carry out this absorption you need to understand that it is called sodium dependent. Means what? Together with glucose, sodium will also get reabsorbed. Now listen, in general sodium is not present inside the cell, present in a very much low quantity. Now in the basolateral membrane, okay, in the basolateral membrane there is a protein like this. It is called sodium potassium pump or sodium potassium ATPase. You know what happens? It has got alpha and beta subunit. Okay, this alpha subunit and beta subunit. Now listen, It's a p-type channel. Now listen with all your attention. Sodium potassium pump or sodium potassium ATPase, what it does is actually it takes out 3 sodium from the cell and allows 2 potassium to enter the cell. Now listen, when sodium potassium ATPase is activated, that means they are using ATP for this throwing of sodium. Why they are using ATP? Because potassium is already abundant in the cell. That's why cell do not need any more potassium, but they will have to take the potassium in because they are throwing sodium out. Now listen, this is actually against the concentration gradient. Why? Because potassium concentration inside the cell is high, so potassium should go from high to low concentration, and sodium's concentration inside the cell, sodium concentration inside the cell is low. Therefore, from intracellular spaces, sodium should enter the cell, but just opposite is occurring. That means it is against concentration gradient. Concentration gradient means what? Concentration gradient means higher to lower. When you are opposing this concept of higher to lower, then it is called what? Then you need ATPases. Reverse concentration gradient. Now listen. So we are throwing excess amount of sodium outside, right? Now listen, when we are throwing excess amount of sodium outside, you should understand one thing, that when you are throwing excess amount of sodium outside, then this sodium, this sodium dependent glucose transporter which is a special protein present in the, which is expressed by the jejunal cells, they will be activated and they will hold sodium here, and in this hand they will hold glucose. Sodium in one hand and glucose in the other hand, and they will just turn inwards. Okay, that means they will now release the sodium and glucose. That's it. They will now release the sodium as well as glucose. Clear? Now listen. Now this sodium again is taken out by this sodium potassium ATPases, and glucose is now entering. Okay. Now listen with all your attention. You know something called galactose? Galactose also uses the same route. That is, with the binding of sodium, glucose or galactose can bind. As the concentration of glucose is more, therefore in most of the cases glucose binds, but SGLT also transfers galactose from outside to inside. Okay, glucose plus galactose. Okay, clear? Now listen. So we have already discussed how glucose and galactose is transferred from outside to inside, that is into the cell. But how will fructose gets absorbed? There is a different mechanism. That is, there is a transmembrane protein called GLUT5. There is a transmembrane protein called GLUT5, and GLUT5 binds with fructose, and when GLUT5 is bound to fructose, it will flip inwards and fructose is released into the cell. Okay, fructose is released into the cell. Now listen. Now this fructose and glucose and galactose will have to leave the cell. Now listen. Almost 80% of the fructose, glucose and galactose which is entering the cell is thrown outside. Okay, and 20% remains for cell's own metabolic purposes. Cell's own metabolic purposes. Now listen. Now this 80% fructose, glucose, galactose will enter the bloodstream. Okay, will enter the bloodstream. Now, how this occurs? Now listen. For the exit of fructose as well as glucose, here GLUT5 is present. Again, interesting part for you people to understand is, okay, sorry, GLUT2 is present. Okay, now listen. So glucose and fructose actually binds with GLUT2, and GLUT2 now flips back, and fructose and glucose is taken by blood. Now you should understand again that glucose also, galactose also follow the same, almost same route. Okay, that means it is also effluxed via GLUT2. That is galactose. Now listen with all your attention possible. Now important point to understand is this. Now our jejunal cells contain SGLT1, okay, right? In PCT also glucose is absorbed, right? Why? Because primary urine or glomerular filter contains some amount of glucose, and glucose is a threshold substance. That means 100% of the glucose is absorbed in the PCT. But how? Listen. Actually, in the PCT or proximal convoluted tubular cells, there is a variation of this SGLT1 protein, which is SGLT2, okay, which is called SGLT2. Now listen. Type 2 diabetes mellitus? Now, a very important insight is this. SGLT2 blockers are used in the treatment of type 2 diabetes mellitus, okay, which is present in proximal convoluted tubule. So SGLT2 is present in proximal convoluted tubule, and SGLT1 is actually present in jejunal cells. Okay, that's all for today. We have successfully discussed the carbohydrate digestion and absorption. Okay.